The fourth type of macromolecules in our body after carbohydrates, lipids, and proteins are nucleic acids. They are named so because of their location in the nucleus and their acidic nature. They are also polymers, meaning that they are also long chains made up of small repeating subunits. In the case of nucleic acids, the small repeating subunits are called nucleotides, which are held together by a phosphodiester linkage. There are two types of nucleic acids in our body: deoxyribonucleic acid or DNA and ribonucleic acid, also known as RNA. They both are very similar. Let's discuss them one by one. A nucleotide is different than any other monomer because it is further composed of three different subunits. First of all, we have a ribose sugar. It is a 5-carbon sugar. Actually, in case of DNA, the oxygen from the OH group at carbon number 2 is removed and we have only hydrogen here. That's why it is known as deoxyribose. The other two subunits of a nucleotide are phosphate group and a nitrogenous base. This hydrogen atom from the nitrogenous base and OH group from ribose react together to give a water molecule, and the two groups are joined together through a covalent bond. Similarly, these two OH groups from phosphate and the carbon number 5 of ribose also react together to give another water molecule, and the two groups are joined together through a covalent bond. When two nucleotides come close together, the OH group at carbon number 3 of the ribose sugar reacts with this OH group of the phosphate group present below it to give a water molecule, and the two groups are joined together. We can see that phosphate group here is linking the two nucleotides through ester linkages. That's why it's known as phosphodiester linkage. Large number of nucleotides are joined together in this way to form long chains. At one end of the chain, there is always a free phosphate group attached to the carbon number 5 of the ribose sugar. That's why it's known as 5' end of the nucleotide chain. At the other end, there is always a free OH group attached to the carbon number 3 of the ribose sugar. That's why it's known as 3' end of the nucleotide chain. The ribose and phosphate groups form the backbone of the nucleotide chains while the nitrogenous bases protrude outwards. A DNA molecule actually consists of two chains of nucleotides which are antiparallel to each other, meaning that this chain will have the 3' end here, and the nucleotides face each other. The two strands of DNA are held together by hydrogen bonds present between the nitrogenous bases. A DNA molecule has four different types of nitrogenous bases. Adenine, also abbreviated as A, and guanine, abbreviated as G, has two-ring structure, while thiamine, abbreviated as T, and cytosine, abbreviated as C, both have single-ring structure. The base pairing in nucleic acids is highly specific. Adenine always pairs with thiamine by 2 hydrogen bonds, while guanine always pairs with cytosine by 3 hydrogen bonds. It's very important to always remember this base pairing, AT and GC. Since the base pairing is always complementary, if we know the nucleotide sequence of one strand of DNA molecule, we can always predict the nucleotide sequence of the complementary strand. Oh, and one more thing. The two strands in the DNA molecule are not just straight, in fact they are wound around each other forming a double helix structure, with nitrogenous bases inside the loops held together by hydrogen bonds as we discussed earlier. Fascinating, isn't it? So this is everything you need to know about DNA. Now let's discuss RNA. Just like DNA, RNA is also made up of repeating subunits called nucleotides, which are in turn made up of a ribose sugar, phosphate group, and a nitrogenous base. In fact, in RNA, we don't have deoxyribose, but the OH group at carbon number 2 is still intact, so we have a simple ribose sugar. Just like DNA, RNA has four different types of nitrogenous bases: adenine, guanine, and cytosine. But in thiamine, this methyl or CH3 group is replaced by a simple hydrogen atom. That's why it's known as uracil or U. Thirdly, RNA does not consist of two strands of nucleotides. In fact, it is made up of a single chain of nucleotides, with ribose and phosphate groups forming the backbone and nitrogenous bases protruding outside. And lastly, unlike DNA, there are three different types of RNAs in our body. Messenger RNA, also known as mRNA, that constitutes 3 to 5% of total RNA in our bodies. Then we have ribosomal RNA, also written as rRNA, that constitutes about 80% of the total RNA in our body. And finally, we have transfer RNA or tRNA, that constitutes about 15% of the total RNA in our body. These different types of RNAs have similar structures but very different functions. In our cytoplasm, we have a cell organelle called ribosome that synthesizes proteins for our body. These ribosomes are made up of ribosomal RNA. DNA is present in our nucleus. The two strands of DNA split and a messenger RNA is formed with one of the strands by complementary base pairing. Note that the sequence of nucleotides in messenger RNA is same as this strand of DNA. The messenger RNA carries this message in the form of the specific sequence of nucleotides to the ribosome in the cytoplasm. Then we have transfer RNA, which has an amino acid attached at one end, and the nucleotide chain forms three loops, which are maintained due to complementary base pairing like this. The three nucleotides present at this location are very important. They vary for each transfer RNA molecule and they pair with their complementary nucleotides present on the messenger RNA. In this way, transfer RNAs bring different amino acids to the ribosomes based on the nucleotide sequence on mRNA. The ribosomes then attach these amino acids, forming long peptide chains. In this way, the nucleotide sequence in DNA is translated into amino acid sequence in proteins.